Hey everyone, and welcome. Today we're going to take a trip deep inside our own bodies to follow the life story of a tiny, but absolutely essential molecule. This thing works tirelessly, every single second, to keep us alive. We are talking about heme. So just think about that for a second. What microscopic machine inside you is running a delivery service that's way more complex and vital than anything in the outside world? Well, you guessed it, that's the hero of our story today.
We are going to follow the entire life cycle of this incredible molecule. We'll start at the very beginning, seeing how it's born. Then we'll look at its life's work. What happens when its job is done? And finally, what happens when the instructions to build it go wrong?
All right, so before we can really appreciate its journey, we need to get a handle on what this hero molecule actually is. Let's start with the basics, the fundamental framework. Okay, so at its core, heme has one very specific job: to grab onto a metal ion and hold it tight. It's built from 4 smaller molecules called pyrrole rings. You can see they kind of look like little pentagons. When these 4 link up, they form a bigger, flatter structure called a porphyrin. And the whole point of this really intricate design is create this perfect, secure little pocket right in the center for that crucial metal ion. And this image shows that structure perfectly. A great way to think about this porphyrin ring is like a molecular chassis, a sturdy frame. And that M right there in the middle, that's the VIP seat. That spot is reserved for its very important passenger. For the heme in our blood, that passenger is iron, and that iron is what gives heme all its power. Now, while we all know heme for its starring role in our blood, it's actually a super versatile molecule that plays a bunch of other parts in the body. This porphyrin structure is just so useful that nature uses it all over the place. It's essential for things called cytochromes, which help generate energy in our cells, and it also forms the active core of enzymes like catalase and peroxidases, which are basically the cell's cleanup crew, neutralizing all sorts of harmful stuff. So every hero has an origin story, right? Well, for him, that story begins inside the powerhouses of our cells, the mitochondria. This is the factory floor where our little molecule gets built, right from scratch. Every complex structure has to start with simple building blocks. And for heme, the whole incredible process kicks off with just 2 basic ingredients: a molecule from our energy cycle called succinyl-CoA, and one of the simplest amino acids out there, glycine. It's pretty amazing that from these 2 humble components, the body builds something so vital. So from those simple ingredients, a super complex, highly regulated assembly line takes over. First, our 2 ingredients combine to form a new building block called ALA. Think of these like individual Lego bricks. Next, a few of these ALA bricks link up to form a chain. Then that chain does this clever little fold onto itself to make the first basic ring structure, and after a few more chemical tweaks, this ring gets converted into its final form before the grand finale, protoporphyrin. And now for the final, most crucial step. The protoporphyrin ring is complete, the chassis is built. The very last thing to do is to slot that critical iron atom right into the center. And with that one action, boom, the molecule is complete. It is now officially heme, and it's ready to get to work. So, with our heme molecule now fully assembled, it is ready for its primary mission. It gets paired up with these special proteins called globins, and together they form the famous molecule that gives our blood its color and, more importantly, delivers oxygen to our entire body. Right, so this combination of heme and globin creates hemoglobin. And honestly, the best way to think about hemoglobin is as your body's personal oxygen taxi service. It's so perfect. It picks up a passenger, oxygen in the lungs, cruises through your bloodstream, and then drops it off exactly where it's needed, in every single tissue, from your brain all the way down to your big toe. But what's really fascinating is that this taxi service actually gets upgraded as we grow. In the womb, a fetus uses something called fetal hemoglobin, or HbF. And this version is especially sticky for oxygen. Why? Well, it has to be able to literally pull oxygen away from the mother's hemoglobin right across the placenta. It's an absolutely brilliant adaptation. Then, after we're born, we switch almost entirely to adult hemoglobin, HbA, and that's the model that serves us for the rest of our lives. But, the red blood cells carrying all this hemoglobin can't work forever. After about 120 days, their shift is over. So what happens then? Does the body just throw it all away? No way. It kicks off one of the most efficient recycling programs you can possibly imagine. When an old red blood cell is ready for retirement, it gets taken apart piece by piece. The globin proteins? They're broken back down into amino acids so they can be used to build new proteins. That valuable iron is carefully pulled out of the heme and sent right back to the bone marrow to create the next generation of red blood cells. But that leaves the porphyrin ring itself, which is now basically a waste product that needs to be safely taken care of.
So that leftover ring gets converted first into a green pigment, then quickly into a yellow pigment called bilirubin. This bilirubin hitches a ride to the liver, which processes it so it can be flushed out of the body. But if the liver can't keep up, maybe because too many red blood cells are breaking down at once, or the liver itself isn't working at 100%, then that yellow bilirubin starts to build up in the blood, and that's what causes jaundice, that visible yellowing of the skin and eyes. It's a direct sign that the recycling pathway is getting overwhelmed.
Now, in newborns, this situation can become really critical. Their livers are still just getting up to speed, which makes them way more prone to high bilirubin levels. And if those levels get dangerously high, the bilirubin can actually cross into the developing brain and cause a very severe but preventable form of brain damage called kernicterus. It's a pretty stark reminder of just how important this whole disposal process really is. So far, we've followed the journey of a perfectly made molecule. But what happens if the genetic blueprint for hemoglobin, the actual instructions for building those globin proteins, has an error in it from the very start?
Well, these genetic mutations can cause two main types of problems with hemoglobin. One type of error leads to a molecule that's structurally messed up. The shape of the protein is just wrong. The other type of error leads to a drop in production. You just don't make enough of it.
A really simple way to think about this is like a factory error. Thalassemia is a production problem. The assembly line just isn't making enough of one of the necessary protein chains. Hemoglobinopathies, on the other hand, are a structural problem. The factory's making the parts, but because of a flaw in the design, they're being built incorrectly.
With thalassemia, the key word is imbalance. If you don't produce enough of, say, the alpha globin chain, then the beta globin chains you do make can't find their proper partners. This sets off a whole cascade of problems inside the red blood cell, leading to abnormal hemoglobin and often really severe anemia. It's fundamentally a disease of quantity. And then with hemoglobinopathies, the most famous example by far is sickle cell disease. Here, a single tiny error in the genetic code for one of the globin chains causes a structural defect. And this one little flaw makes the hemoglobin molecule stick together under certain conditions, warping the entire red blood cell into that rigid sickle shape. This is a disease of quality, where the shape of the molecule changes literally everything. And that brings us to the end of our journey. We started with just two simple molecules. We followed them through this complex assembly line to create a perfect oxygen carrier. We watched this elegant recycling system at work, and we saw how one single error in the blueprint can have catastrophic results. It really makes you think, doesn't it? What does this incredible balance between molecular precision and profound vulnerability tell us about the fragility and the amazing resilience of life itself?